Hey everyone, I hope you're having a lovely day, and I am here to present my presentation on Agrobacterium and the genetic modification of plants. So you're probably thinking to yourself, what is Agrobacterium? Agrobacterium is a bacterium to whom we owe a great deal of our current diet. It was first described by H.J. Kahn in 1942, and it has been classified in the genus of a gram-negative bacterium. This bacterium is commonly found throughout the world in the soil and is transported onto the plants via dust in the wind or splashes from rainfall. Its main characteristic, and the characteristic that makes it so vulnerable, is its ability to transfer DNA or its own unique thumbprint to plants by attaching to any defect or wound that might be exposed on a plant or its roots. Since its first discovery, Agrobacterium has been studied for over 100 years and has been revolutionized plant molecular genetics and the advent of genetically modified crops. It has literally changed the way we feed the world and is currently used by most major agricultural growers to improve their crops. When modifying anything from its originally designed natural condition, it is important to ask if the modification is necessary. Why should we encourage this modification? One only has to look at the current population growth against the diminishing usable land available for planting crops and grazing livestock. When global warming is added to the picture and sea levels rise, reducing the land even further, it does not take a scientist to figure out we are headed for a worldwide food shortage. With the advent of genetic modification at the cell level in plants, farmers have been granted a whole new way of ensuring their crops are healthy, sustainable, and transport ready for a world yearning to buy their crops. Genetic modification allows crops to be altered to increase their resistance to insects, increase their shelf life and stability, increase their tolerance to heat and cold during transport, and even yield higher outputs. Agrobacterium has the unique quality of being able to transfer DNA to a plant through a gene sequence. Prior to Kahn's discovery in 1942, tumors or galls were first identified and recorded on plants in 1679 by Marcello Malpighi, an Italian biologist and physician. Malpighi is referred to as the founder of microscopical anatomy, histology, and the father of physiology and embryology. These tumors were studied in 1897 by Fridono Carvier from Naples, Italy. Was the first to isolate Agrobacterium. His work resulted in the first recognition of the plant tumor disease known as crown gall disease. It occurs on both woody and plants, including fruits and flowers. When Agrobacterium attaches to a wound or plant sore, it initiates the growth of a tumor. It does this by transferring its own DNA into the cell structure. Scientist Armin Braun developed a concept called the tumor-inducing principle, also known as TIP. With Agrobacterium, which has the unusual nutritional property of being able to start an infection without being necessary for the growth of the infection, genomes of plants and fungi can be engineered by the use of Agrobacterium for the delivery of sequences hosted in the transferred DNA, binary vectors. The tDNA is transferred from the bacterium into the host plant's nuclear DNA genome. The tDNA is surrounded by 25 base pair repeats on both ends. The transfer begins at the right end and is destroyed at the left end. After the transferring of the tDNA into the plant's genome, the bacterium reconstructs the plant cells to grow into a tumor and produce the food source for the bacteria. How exactly does this transfer happen? Agrobacterium contains Ti plasmid, which can induce tumors and is separated into different sections based on its type of molecule. The TI plasmid is an independent replicating DNA molecule that is inside Agrobacterium cells. The TI plasmid has three regions, which are tDNA region, virulence region, and open catabolism region. The tDNA region has borders that are set of 25 BP border sequences present on either sides of the tDNA and are transferred to plant cells. The right border is usually more crucial for the tDNA transfer. The virulence region is where genes are responsible for the transfer of the tDNA into the host plant, and they are located outside the tDNA. The open catabolism region looks for proteins that are involved in the uptake of the opens. Agrobacterium originally was divided into three types dependent on the way in which it attaches and affects the target plant. The most common and most utilized version is Agrobacterium tumefaciens, which results in crown gall disease. Agrobacterium rhizogenes manifests in the roots of its target plant, causing hairy root disease. And lastly, Agrobacterium radiobacter, similar to the tumefaciens, also may attach to cells both inside and outside of its target plant, resulting in virulent strain. The technological advances stemming from the identification and study of Agrobacterium have been countless. Advances in plant gene function, cell biology, plant microbe interaction, and biotechnology have been made. Many crops worldwide use techniques associated with Agrobacterium in the production of maize, rice, barley, wheat, and many more staple crops. Agrobacterium is an incredible organism that has very unique genetics and functions. It is very helpful to the surrounding world, and more studies should be researched about it. Thank you for coming and listening to my presentation, and I hope you guys have a wonderful day. Thank you.